talks are covering a wide range of topics that are related to the infection in CF airways. If you haven't had a look at their poster so far, I would strongly encourage you to do so. There's a little bit of housekeeping just before we start the session. When you look at your screen, you'll see on the right-hand side a text box with two separate tabs on them. One is for chat and one is for the question and answer sessions. So please feel free to chat socially in the chat tab. But if you would like a question to be seen by the moderator and the speakers, please add that to the question and answer tab, and then that will get through to the moderator for the question session after each speaker's presentation. Okay, so our first talk is from Dr. Paula Zamora, who's from the University of Pittsburgh, and she's going to be talking to us about her research studying Pseudomonas aeruginosa bacteriophage and its interactions with host respiratory cells. So thank you, Paula.
Hi, everyone. Thanks for that introduction. I'm very excited to share with you some of my work in the Bomberger lab, where I'm trying to understand how phages that are used in phage therapy influence cells of the respiratory tract. And I don't have any relationships to disclose. In the Bomberger lab, we focus most of our work in Pseudomonas aeruginosa infections. And as you're probably aware, Pseudomonas is increasingly becoming antibiotic resistant, and alternative therapeutic options are urgently needed. One of these options is phage therapy, which relies on the use of viruses that kill bacteria. Phage therapy has been used successfully in CF to treat multi-drug resistant Pseudomonas infections. And I want to highlight the work from our collaborators, John Koff, Ben Chan, and Paul Turner from Yale University. In their study, they recruited 10 individuals with CF that had been diagnosed with chronic Pseudomonas infection. And they treated them with a combination of 3 lytic phages, OMK01, TIVPH6, and LPS5. These phages were administered in a nebulized form. After treatment, they observed an increase in lung function as well as a decrease in bacterial counts, indicated that phage therapy is a feasible alternative to treat chronic Pseudomonas infections in CF. From these 3 phages, I have focused most of my work in one of them, OMK01. OMK01 is a phage that has broad strain specificity, meaning that it can kill several Pseudomonas clinical isolate strains in vitro. OMK01 is a myovirus with a large double-stranded DNA genome, and during infection, phage OMK01 binds its specific receptor, depositing OPRM on the surface of Pseudomonas. The OPRM makes a B system. It's an efflux pump that is responsible for pumping out several classes of antibiotics. When Pseudomonas expresses this system, it becomes antibiotic resistant, but at the same time, it becomes OMK01 sensitive as the phage has its receptor to bind. As a result, OMK01 forces a genetic trade-off in which antibiotics and phages can act together to ensure that they effectively eliminate Pseudomonas. This makes OMK01 a great therapeutic. Despite that phage therapy is a successful alternative to traditional antimicrobials, the effect of high doses of foreign phages, like the doses that are used therapeutically, on the human host has not been extensively studied. And to address this gap in knowledge, we came up with 3 questions to answer in my studies. Do phages used in CF phage therapy enter cells of the respiratory tract? Do these phages elicit innate immune responses? And what is the effect of CFTR modulator therapy on phage mammalian cell interactions? In this talk, I'm only going to show progress on the first 2 questions. In my research, we use a model that is based on bronchial epithelial cells that are grown in transwell inserts. In this model, cells are grown at air-liquid interface, which allows them to polarize and develop apical and basolateral sites that mimic interactions with the lumen of the airways and the blood, respectively. To determine whether phage treatment in our model mimics the reduction in bacterial burden from the Yale study, we conducted an assay in which we inoculated our cells with OMK01 and Pseudomonas for 1 hour and measured bacterial attachment. In this experiment, we observed a 2-log reduction in CFUs just after 1 hour treatment. We also conducted a similar assay in which we inoculated cells first with Pseudomonas, let it form biofilms, and then we came with OMK01. And we measured how in 2 hours CFU reduction after OMK01 treatment. In this experiment, we observed a 1.5 log reduction in bacterial counts in the 2 hours that OMK01 was present. This 1.5 log reduction is similar to the amount of bacteria that we detect after we treat cells with antibiotics. These studies suggest that OMK1 is able to effectively kill Pseudomonas in our bronchial epithelial cell model. To understand how phages used in phage therapy interact with the airways, we decided to first look at where OMK1 enters bronchial epithelial cells. And understanding phage entry, and more importantly, knowing where phages localize subcellularly after they enter, will give us important information about potential intracellular innate immune pathways that phages could be activating. To study phage entry into mammalian cells, we first wanted to look at whether phages could cross the epithelial apical membrane. And to do this, we treated cells with fluorescently labeled OMK01, that is shown in green here, and conducted confocal microscopy experiments in which we stained the apical membrane with dermagglutinin, that it's a lectin that binds the glycocalyx, and it's shown here in red. Here you can appreciate the green particles located underneath the red glycocalyx, which is better appreciated in this video. These results suggest that OMK01 can enter bronchial epithelial cells. Considering the important role of ciliated cells in mammalian viral infections, we then wanted to look at whether there was any co-localization between cilia and OMK01. And we conducted similar experiments as the one I just described, and to look at cilia, we stained cells with antibodies specific for acetylated alpha-tubulin, that it's a protein that is highly expressed in the cilia of airway epithelial cells. In these experiments, we detected 2 main populations of phages, OMK01 that associates with cilia, that is shown in yellow, and OMK01 that localizes in discrete compartments inside the cells. You can also see these populations here. We decided to investigate further these populations that appears to localize in discrete intracellular compartments, because we are interested in potential pattern recognition receptors that could be activated by these phages. In similar experiments, we conducted confocal microscopy after phage treatment and labeled the following organelles: early and late endosomes, autophagosomes, lysosomes, and the endoplasmic reticulum and Golgi, using antibodies specific for the proteins that are indicated here. In these experiments, we detected co-localization with the 4 vesicular organelles that we tested, suggesting that OMK01 can actually be trafficked through the endosomal system. We did not observe co-localization with the endoplasmic reticulum markers or the Golgi markers, suggesting that phages do not reach these organelles. As we detected co-localization with the lysosomes, we wanted to better understand what was the degradation kinetics of OMK01 inside our bronchial epithelial cells. So in this experiment, we treated cells with phages and then measured intracellular phages over time. And we detected that by 48 hours, all intracellular phages had been degraded. Within this time course, we also did not detect any phages in the basolateral compartment. And it's important to mention that in this experiment, or in any of our experiments, we did not observe a decrease in transepithelial electrical resistance, suggesting that epithelial integrity is maintained after OMK01 treatment. As I mentioned earlier, we're very interested in determining whether phages can activate innate immune responses. And to test that, we treated cells with OMK01 and measured the antiviral cytokine interferon lambda at 24 hours post-treatment in both the apical and basolateral compartment. we observe an MOI-dependent increase in basolateral interferon lambda secretion over time, suggesting that OMK1 can induce an antiviral response. We also look at pro-inflammatory cytokines, and this is very preliminary data, but by 24 hours, we did not observe a change in IL-1 beta secretion. But when we went and look at 48 hours, we observe a 75% decrease in basolateral IL-1 beta. We also screened for other pro-inflammatory cytokines like IL-6, and we did not observe a difference after phage treatment. And future experiments will aim to elucidate the role between interferons and IL-1 beta in OMK1 immune responses. The main takeaways from my talk are that Pseudomonas phage OMK1 can enter bronchial epithelial cells and colocalizes with cilia and endocytic organelles. OMK1 is degraded within 48 hours and does not cross to the basolateral space, and OMK1 appears to induce antiviral immune responses. Future experiments will be focused in determining phage entry mechanisms and immune responses using phages from different families. We also are planning to assess cytokine secretion profiles in sputums from individuals with CF that have received phage therapy. We're also interested in determining how CFTR modulators can change phage entry mechanisms and immune responses. And finally, we want to evaluate the effect of the cytokine secretion after OMK1 treatment on infections with respiratory viruses and phage-resistant Pseudomonas strains. And with that, I would like to acknowledge everyone in the Bomberger Lab, our collaborators, funding sources, and thank you for your attention, and I will take any questions that you may have.
Thank you, Paula. That was awesome. Some of those confocal images are awesome. And I'm a big fan of confocal microscopy. We have one question to start with. So what is the nature of the association between your phage OMK01 and cilia? And are these interactions also seen at the altered pH values tested?
So we do not know yet. So there are two things. So in our experiments, we do extensive washing to try to remove as much phage as we can, but we haven't assessed if that phage is first localized extracellularly or intracellular. They could be reminiscent phage. So some experiments that we're planning to do is doing acid washes, for example, to remove as much phage as we can to really kind of figure it out how is the interaction between the cilia and the phages. And what was the other question? I'm sorry.
Are these interactions also seen at the altered pH values tested? So I guess, again, you'll...
Yeah, so we don't know yet. So those were at the acidic pH that we tested because that's the... so I didn't show it. So if you go to my poster, you can see it. I didn't show it here. But we see that there is a pH range in which phage entry seems to be enhanced. So all those experiments were done within that pH range.
Okay, so another question is there a difference in the number of intracellular phage between healthy and CF cells?
That's a very interesting question, and we don't know yet. So we do, it's in the, we want to test that. We do see that there is a pH dependent mechanism for entry, and acidic pH seems to facilitate entry. We don't know how, and considering that CF cells have an acidic air-liquid interface compared to healthy cells, we do think that there could be a difference related to that. We also think, we haven't done these experiments yet, that mucus viscosity might also affect how phages interact with potential cellular receptors. So all those things could be involved, but we haven't tested it yet.
Okay. Another question from Anonymous. Very nice work. Do you know whether these phage occur naturally in the Pseudomonas aeruginosa infected airway?
Oh, that we don't know yet. So I think that those experiments are going to come from probably work from Paul Turner's group, that they actually have the cohort and they are doing whole genome sequencing and microbiome studies from those individuals with CF.
That actually lets me sneak in a question kind of related to that. Thinking about the role of prophage. So obviously you're adding in these phage exogenously as a treatment. And if you think that there may be some, I guess, lysogeny, where prophage in the genome of Pseudomonas are released as a result of being in contact with host cells, and maybe that might play a role with your phenotype, and if there's a way that you can maybe potentially test that and see if
that is a really a good point that I think it also will kind of be elucidated from the studies that Paul Turner's group is doing. In all my experiments or in most of my experiments, I haven't yet put the bacteria. So the only experiments that I showed today that actually have biofilms on top are the experiments that I was measuring the kinetics of phage bacterial killing, but not phage entry into the cells. So I think those are good points that once this project moves a little bit forward, we will be able to kind of combine everything together and test some of those questions.
Awesome, thank you. I think there's a couple more questions in the Slido chat, so if you have time to answer that, that would be great. And otherwise, we'll move on to our next speaker. Thank you, Paula. So our next speaker is Dr. Frank Robledo Avila. Hopefully I've pronounced your name roughly right. And Frank is from Nationwide Children's Hospital in Ohio. And he's going to be talking to us today about the FTR modulators and their effects on neutrophils in the CF airways. Okay, thank you.
Thank you to the organizers to give me the opportunity to present my research in this meeting. Today, we are going to talk about how CFTR modulators impact in the antimicrobial functions of CF neutrophils. There are no relationships to disclose related with my presentation. Just a quick reminder, cystic fibrosis is the most common genetic disease and remains with no cure. CF is caused by mutations in the CFTR ion channel, affecting several organs, including lungs. The CFTR dysfunctions induce physiological changes promoting the establishment of bacterial infections. This process induces a recruitment of inflammatory cells, mainly characterized by the accumulation of neutrophils. These cells are unable to clear the infection, but promote the lung degeneration. Since CF was clinically described, different types of therapies have been released, increasing the life expectancy of CF patients, being the antibiotics one of the main treatments that help dramatically the CF patients. However, during the last decades, have been more common to find multi-drug resistant bacteria in the lung of patients. Fortunately, in recent years, a new type of drugs were released called CFTR modulators. These molecules aim to target the CFTR channel to improving the function of it. This therapy is obviously the new hope for the CF patients. So the CFTR modulators, we can divide it into two types: the potentiators and the correctors. The potentiators increase the function of the CFTR channel, like ivacaftor, for example, and the correctors help CFTR protein to reach the plasma membrane to the cells, increasing the density of the channel in the cells, like lumacaftor or tisacaftor. And the combination of both help to increase the density of the receptor in the cells and also the function of the CFTR channel. Most of the studies related with the CFTR modulators and the description of these drugs were performed in lung epithelial cells, and the mechanisms are known at this point. However, we don't know exactly how these modulators are going to affect the immune response. And due that the phagocytes are critical during the inflammatory phase in the lungs of these patients, we wanted to know the interaction of these drugs with the phagocytes. Some previous reports in the literature show that either macrophage or neutrophils are affected positively in the different cell functions. Macrophages increase some antimicrobial killing against some bacteria, and also neutrophils are having a positive effect with some modulators. So based in this previous data, we wanted to explore more in detail. First, obviously, there are several questions about phagocytes and CF, but we wanted to explore first if phagocytes express functional CFTR ion channel. because there is some controversy about this. So for doing this, we perform a whole cell patch clamp analysis. So first, here you can see in the left panel, we use CF macrophage and healthy macrophage. So basically what we did is we measured the current of the CFTR channel. As you can see here in the black line, the current expressed of the CFTR expressed in macrophage, healthy macrophage. And when we add an inhibitor of the CFTR channel, as you can see here in the blue line, the current was totally blocked. We also tested that current in CF macrophage, and we saw a very reduced current, almost absent. So next, what we did is basically we collect neutrophils, healthy neutrophils, and we tested the CFTR current in these cells. We saw some current of CFTR, and the current was partially inhibited when we added an inhibitor of the CFTR channel. Additionally, we wanted to know if the CFTR modulators, in this case, a combination of ivacaftor, lumacaftor, could potentiate the CFTR current in neutrophils. As you can see here in the right graph, after the treatment, the neutrophils expressed a higher current of CFTR in the cells, very similar as macrophages, as you can see, suggesting that indeed the CFTR modulators are improving increasing the current of the CFTR channel in the cells. Based on this, our next question was if the CFTR modulators could modify the expression and the localization of the CFTR protein in neutrophils. So we isolate healthy neutrophils and neutrophils from CF patients, and we treat the cells with single therapy with lumacaftor or tisacaftor. These experiments were done in vitro, and we stain the cells with anti-CFTR. As you can see here, with healthy cells, with healthy neutrophils, tisacaftor helped to increase the density of the intensity of fluorescence for CFTR in neutrophils. We saw similar results with CF neutrophils. Basically, the intensity of fluorescence for CFTR staining increased dramatically. Then we also tested the combination of different drugs. For example, ivacaftor, lumacaftor, ivacaftor, tisacaftor, or the triple combination. And we also measured the intensity of fluorescence for CFTR. We saw very similar results as the single combination. Healthy neutrophils increase the density of the channel in the cells, very similar with the CF neutrophils. Our next question was how these changes in the expression and the function of the CFTR channel induced by the modulators affect the antimicrobial mechanism of CF neutrophils, which is their primary duty. We collect CF neutrophils and healthy neutrophils, and we infect the cells with Burkholderia cenocepacia, which is a CF pathogen. After 3 hours of the challenge, we measured the antimicrobial killing. As you can see here, 100% correspond to our controls, the healthy neutrophils. So the CF neutrophils show a reduced antimicrobial killing compared to healthy cells. We also isolated neutrophils from patients on the treatment with Symdeko 3 months after they started the treatment. So we didn't see changes in the antimicrobial killing in these neutrophils. But we wanted to make sure that the drugs could affect or interact more in detail with the cells, neutrophils. So what we did is basically we isolated neutrophils, and then we treat the cells in vitro with a single treatment of lumacaftor, tisacaftor, or the combination of ivacaftor, lumacaftor, or ivacaftor, tisacaftor. Very interestingly, we saw an increase in the antimicrobial killing in CF neutrophils treated with any of the modulators, suggesting that indeed the CFTR modulators are potentiating the antimicrobial mechanism of the cells. And very similar results we saw that with a double combination of the modulators. but how the CFTR modulators are potentiating the antimicrobial functions of neutrophils. So we were interested to measure the oxidative burst in neutrophils because it's one of the main mechanisms of these cells to kill bacteria. What we did, basically, we treat the cells with any of the CFTR modulators, and we measure the oxidative burst, the spontaneous oxidative burst of these cells. In the left panel, you can see the response of healthy neutrophils, and after the treatment with the modulators, the cells produce high amounts of ROS species. With the CF neutrophils, even that they produce less ROS as compared as healthy neutrophils, they were able to produce ROS after they were exposed to the CFTR modulators. Additionally, because there are several antimicrobial mechanisms derived of the ROS, but also independent of ROS, we wanted to explore another mechanism of neutrophils. One of the most important mechanisms of neutrophils is the production of extracellular traps or neutrophil extracellular traps, which is the last antimicrobial mechanism of neutrophils until they die. So we isolate neutrophils, and then we stimulate the cells with PMA for 3 hours. And at the same time, we treat the cells with any of the CFTR modulators. With what we saw, and we stain the cells with anti-DNA and neutrophil elastase. As you can see here, non-CF cells produce a large amount of nets as compared to the CF neutrophils, which we can see some cell death, but not too many traps at this time point. But after the treatment with any of the CFTR modulators, we saw a potentiation of the production of nets in any of the cells, healthy or CF neutrophils. And finally, our conclusions of this, we are still doing some experiments about this project, but at this point, we know that the CFTR channel is expressed in neutrophils, is functional, and also the CFTR modulators can potentiate the function and the antimicrobial mechanism in these cells. And finally, I just want to thank the people involved in this project. Thank you.
Thanks, Frank. That was a really great talk. I was going to ask a question about nets, but then you already touched on that on one of your last slides, so that was really cool to see that. I did have one question to start off. Was the localization of CFTR the same in non-CF and CF cells when modified with correctors?
So we see an increased density of CFTR staining in CF neutrophils. However, we don't see the same levels. So it's still a difference, and obviously it depends on the patients. Sometimes we see, well, the primary cells, you know that the behavior changes depending on the donor, but we see a very, high amount of receptors in the cells. Almost the same. I will say almost after the treatment, they have similar intensity of fluorescence. Obviously we will do more tests. We want to do like a flow cytometry with these cells to actually measure the intensity of fluorescence after the treatment.
Awesome. What is the antimicrobial effect of the CFTR modulators? Are they still present in the in vitro assays described in figures 5 of your poster?
So we try to keep the modulators all the time with the cells because we wanted to make sure that the modulators are actually stimulating the cells. So I don't think they have any effect directly to the bacteria, but we don't know. But we still need to test that. Yeah.
Okay, let's see. Got some other questions here on Slido. Do you think stimulating PMN with PMA to measure nets is relevant at all rather than live bacteria?
there is a lot of also controversy about how nets are actually helping the host to kill bacteria, because depending on the type of bacteria, some nets are not able to actually kill bacteria, and they are actually promoting the establishment of biofilms, for example, and sometimes even the dissemination to other cells like macrophage.
Yeah.
So we tested that mechanism because it's still very important for like neutrophils to stop the bacterial infections, but we don't think that is the most effective mechanism against bacteria. And we have approved in CF patients that they accumulate with the time a lot of DNA in the airways, and that promotes the establishment of bacterial infections.
Okay, thank you. This is a good question. What happens when you look at neutrophils or macrophages, from CF patients who've been treated with CFTR modulators? I guess I'm not sure if it was on your poster, but I missed it in the slides, whether the neutrophils you got from your CF blood, if those patients had previously seen the modulators at all.
Yeah, so we have some preliminary results showing that, for example, the triple combination, it's actually increasing the density of the CFTR channel in the neutrophils. So it's helping the neutrophils, it's targeting the neutrophils. And also the antimicrobial killing is potentiated. Not as much as when we treat the cells in vitro, but we see an effect. For some reason, when we were isolating neutrophils from patients on treatment with, for example, tisacaftor, we didn't see effects. I don't know if that's because the life of the neutrophils is very short, because they are constantly exchanged with the bone marrow, and they lose that the concentration that is affecting the CFTR in the cells.
Okay, so I think we've got time for one more question. Were there changes in cytokine secretion profile and or overall levels from Burkholderia infected neutrophils with the addition of a modulator?
So we haven't measured the ability to produce cytokines in these cells, but we know that, for example, macrophage, when they are infected with Burkholderia, they produce a lot of inflammatory cytokines, particularly IL-1 beta, and CF macrophage produce even more than healthy. We haven't measured the production of cytokines, particularly infecting the cell with Burkholderia, but we think that indeed the CF neutrophils they have a more inflammatory profile as compared as healthy neutrophils.
Okay, awesome. Thank you, Frank. I think there's a couple more questions maybe in the Slido chat for you to answer, but otherwise we'll move on to our final speaker of the session. And that is Lauren Gonzalez, and she is from University of Washington. And today she's going to talk to us about antimicrobial susceptibility testing, which I know is a big topic in CF, but looking at it at more of a community level. So thank you, Lauren.
Good morning. Thank you for that introduction. Today, as Dr. Darch mentioned, I'm going to be talking to you about some of our work with community level antibiotic susceptibility testing. Before I begin, I want to thank the organizers for setting up this exceptional virtual event and inviting me to speak. I have no relationships to disclose. So before we dive in, I think it's important to address a key question that's central to the work I'm going to be talking about today, and that is why is the sputum microbiota, or the collection of microorganisms residing in sputum, so important? So in each and every one of us, the lungs are a very important site of interaction with the environment. And when it comes to people with CF in particular, lung disease is largely driven by infection. So not only is sputum readily available, usually, and easier to collect than other methods, but it's also right at that lung environment interface, and it's an important specimen for identifying pathogens, choosing antibiotics when they're deemed absolutely necessary, and studying inflammation. So for the talk today, I'm going to be focusing on these first 2 points. When it comes to respiratory infections and identifying pathogens and antibiotic selection, antibiotic susceptibility testing really guides this process. I'll be referring to antibiotic susceptibility testing as AST. So how exactly does this work? Well, AST begins with the collection of a patient sample, and in our case, sputum. This culture is then cultured on selective media for standard pathogens, and from there, selected representative isolates are then tested in vitro for susceptibility, either through diffusion methods or broth dilution methods. Based on susceptibility results, if antibiotics are required, a treatment regimen is chosen. So AST is a standard clinical laboratory method that's invaluable for treatment beyond just CF, but we find ourselves running into an issue. There's a growing body of literature that's showing that antibiotics selected using standard AST do not reliably lead to clinical improvement. Studies have described that with treatment, no correlation exists between the in vitro susceptibility of standard pathogens that we screen for and the clinical response of the patient. So when we look at changes in lung function and time to next exacerbation, these two were not associated with antibiotic susceptibility. Additionally, some people with CF don't have detectable standard CF pathogens, yet are still admitted for pulmonary exacerbations and respond to treatment. So why do we see this disconnect? Well, if we take a step back and we look at our current AST methods, we find that we only test a few representative isolates, that these isolates are tested in pure culture, and they're tested under very standardized conditions. So a single atmospheric condition, a very specific medium is used, and a very specific inoculum level is used. So looking at this, we see that certain aspects of lung infection and the CF lung itself are not accounted for. For example, not only do diverse susceptibilities exist within pathogen populations of an infected individual, but CF-associated microbes don't exist in pure communities in the lung. Additionally, the lung is not homogeneous. There exist physicochemical gradients throughout the airways. So how do we make what we're currently doing better? We decided to focus on accounting for some of these aspects. So by accounting for oxygen tension, medium content, the presence of other bacteria, we hypothesized that through community level susceptibility testing, we'd be able to see how specific traditional pathogens, as well as the total microbial community, respond to antibiotics, and that it will ultimately be a better reflection of sputum microbiological changes. So here's how we approached this. Our assay started with a homogenized sputum sample. So this sample comes from a cohort. These samples were previously characterized through both culture and molecular methods, and this really set us up to compare our in vitro data to the in vivo data that we have. The sample was then inoculated in a modified susceptibility testing medium in a 96-well plate in the same manner as a traditional AST over increasing concentrations of a selected antibiotic. This process was then done in 3 different atmospheric conditions: aerobic, microaerobic, and anaerobic. Following incubation, we then determined the community minimum inhibitory concentration, or the MIC. That's the concentration at which no visible bacterial growth occurs in the well. We also determined the community bactericidal the minimum bactericidal concentration, or the MBC, and that's the concentration in which no plated bacterial growth. For the MBC, we focused on the pathogens Staphylococcus aureus and Pseudomonas aeruginosa. Lastly, we determined the community composition through 16S amplicon sequencing and the total bacterial load of both the sample as well as certain taxa through qPCR. So because tobramycin is one of the most common used antibiotics in CF care, and it was the antibiotic used for the cohort that the samples originated from, we use tobramycin here as well at a concentration ranging from 0 to 1024 micrograms per mil. And up until this point, we've used 3 previously characterized sputum samples in this assay, but for time constraints today, I'm only going to show you one. So here I'm showing you the culture data that we've collected from this sample because it's really easy to get lost in the numbers. There's a few major points that I want you to take away. The first is that as oxygen presence diminishes, so as we move from aerobic conditions to anaerobic conditions, the susceptibility of both the community as well as specific pathogens decreases. The second point that I want you to take away is that relative to representative isolates from this sample tested through traditional AST methods under the same conditions, pathogens tested in our community level susceptibility assay, when it comes to staph, remain unchanged, whereas susceptibility of Pseudomonas aeruginosa relative to traditional AST increases. When we look at our molecular data, we start to see a bigger picture. So on the top, we have our 16S amplicon sequencing data, relative abundance over increasing tobramycin concentrations. And on the bottom, we have our qPCR data over those same increasing concentrations. A few key things I want to point out. The arrows and these circles represent the culture data that I have just shown you. In blue is Pseudomonas aeruginosa. In this lime green is Prevotella species. In darker green is Staphylococcus aureus. In pink is our Streptococcus species. And in black is the total bacterial load of the sample. I'm showing you, the aerobic conditions here. So a couple things that we noticed was that the culture data, again, I previously showed you, actually corresponds relatively well to both the relative abundance data, so the absences in our organisms at specific concentrations, as well as dips in levels in our qPCR data. On top of that, another thing we noticed is that while Streptococcus species seems to increase in relative abundance via amplicon sequencing, it actually remains relatively steady and closely follows the total bacterial load line until we hit the community MIC, in which we see a pretty substantial drop. So when we compare our aerobic conditions to our anaerobic conditions, we start to see re-emerging patterns. Again, the culture data largely corresponds to dips we see in certain taxa. And additionally, we see that Streptococcus again follows the total bacterial load line. But we do see a couple new things. Those are that we see an increase in Veillonella as well as Prevotella, which makes sense considering the anaerobic conditions. And that's reflected in the qPCR data as well. So just to kind of summarize our major takeaway points. So community susceptibility decreased as oxygen availability diminished, and that was something we saw occur also with the specific pathogens we tested for. The community MICs were largely associated with Streptococcus abundance. Prevotella abundance increased under anaerobic conditions relative to aerobic conditions. And in the presence of the sputum microbiota relative to isolates tested in traditional AST, we find that Staphylococcus aureus susceptibility remains unchanged, while Pseudomonas susceptibility increased. And we find that it's possibly underrepresented in our data, both culture and molecular. So what have we learned so far? Well, we have observed that pathogens do indeed behave differently in the context of the community, and we believe that community-level susceptibility testing gets us closer to creating a framework that allows us to test this more comprehensively and understand the impacts this may have clinically. But I also want to take a moment to recognize that our method still requires some work. We need to address the competitive disadvantage that Pseudomonas is in our modified medium, as well as the competitive advantage that we see Streptococcus might have. Additionally, some taxa consistently observed in the original sample data, which I did not show you here today, was not detected under incubation and after incubation in our modified medium, one of those being Rothia. So ultimately, our method requires further optimization and validation for its potential clinical utility and efficacy. This project was truly a team effort and would not have been done without the investment of the Hoffman Lab, particularly Maria Nelson and Dan Walter. This was pre-COVID times when we could all stand within 6 feet of each other. I also want to acknowledge our funding from CFRI, as well as the Cystic Fibrosis Foundation supported RDP, both of which were essential to the work I've shown you here today. And lastly, I want to thank you all for your time and attention. I'm more than happy to take any questions.
Great. Thank you, Lauren. That was a great talk. antibiotic susceptibility testing is close to my heart. I did some studies on that during my own PhD. So it's really interesting to see what other people are doing. We have a question to start off with. Do you think that the 2 to 4-fold increase in your community MIC that you observed for tobramycin would be similar or the same if you used other antibiotics?
That's a really good question. We have not tested other antibiotics. I do expect, I hypothesize that we would see a decrease in susceptibility as oxygen diminishes. But again, that depends on the antibiotic and whether or not moving from an aerobic state to an anaerobic state impacts its effect on the bugs.
Okay, thank you. We have some questions that are based more on, I think, just a little bit more background on the modified medium that you're using. Can you tell us a little bit more about that?
Yeah, so Dan Walter, I have to give credit where credit's due. Dan Walter really designed this. It's a TSB base with yeast extract, and it's got a lot of the things we find in sputum, hemin, metadione. We used an antifungal as well as beta NAD and nitrate. So it's a rich medium, and Dan designed it to ensure that we could grow as many organisms as possible in it.
Okay, so trying to recapitulate that infection site a little bit more than just the regular, I guess, Muller Hinton broth that's normally used in diagnostics.
Yeah.
So the cohort of samples that you were talking about, are those patients, are they in exacerbation or are they relatively clinically stable? Is there any information you have on those?
I don't know off the top of my head, but this specific cohort was, at least the samples we used were from they were all on tobramycin, and so we were comparing. Maria did this work. She was comparing baseline levels, organism levels to week 1 through week 4 of tobramycin therapy. And so the samples we used were at that baseline to week 1 portion. So I'm not entirely sure about exacerbations. I want to say they were relatively stable.
Okay, awesome. And one final question from Boo Shan Singh. She would like to know, since that PA has a disadvantage and strep has an advantage in the modified media, do you think part of this is due to the interaction between the two? And she mentions Jessica Schofield's talk yesterday.
Yeah, I also attended that talk. So a while back, I actually happened upon one of Dr. Schofield's papers, and it's possible. We haven't started to parse out what exactly is going on with Pseudomonas, but that's definitely one of the potential issues, is that just the sheer amount of Streptococcus there could actually perhaps and be antagonizing Pseudomonas. So yeah, that's one of the things we'd like to really look at.
So many things to do. That's so great to think about when you have these question and answer sessions.
Yeah.
Okay, so we're coming to the end of the session. So I am going to just thank all of our speakers once again for their fantastic talks. Thank you to my co-chair for doing such a great job of moderating the questions for me to ask as we went along. And thank you so much, everyone, for joining us today. The Slido session is going to remain open if you have more questions, but I'm sure the speakers would not mind if you felt you wanted to reach out and ask them questions through an email or the networking tool that you see on the NACFC website. So thank you very much, everyone, and I hope everyone stays safe and has a great weekend.
you.